how's your day today? Good. Good to arrive.
Yeah.
Can I sit on my lap? Okay. Can I have one? You need me?
I'm getting out.
You getting out? What's your vibe? Here, you want to switch me?
How
It's not as hot up here.
is it?
And better?
That's splash you.
You didn't splash me.
That's too cold.
Mm-hmm.
Who knew that? The cells?
Mm-hmm.
Oh man.
The shells.
Yeah, the cells did say. Oh my goodness. And the was up there.
Mm-hmm.
And the cells was on the bowls. Mom,
Mm-hmm.
that's white.
Yeah. You gotta clip your fingernails. How long they are.
Your bubble.
Wash your face off. It's a mess.
Take an eye cloth.
Can you hear me now?
Yes,
Oh, perfect. Okay. Can you see me?
yes, I can see.
Okay, perfect. All right. Let's see here. So there were a handful of people who have not taken the first lab quest when I got online last night. And I don't know if those people are no longer in the class. I know what at least one or two of the names didn't look that familiar to me. If you have not taken it, please email me and let me know. If you don't take those, I promise you, you will fail the lab portion of the lab. And I don't know what else to say except you have to take them.
Yeah.
The high was a 25 out of 25. The low, I think, was an 18. And if you got somewhere, let's say, for example, you got 18.78, you got a 19 because I'm going to round it up. But I didn't want to round it up until after everyone had had their 2 attempts. And so
When I switch to hands.
I'm going to have someone. Any questions for me right now?
No, that was nice that you let it open early. Thank you.
Well, and I think I'm going to do that with this one. I think the one that is for this lab and last week's lab is if I remember right, bear with me, I'm going to actually just look really quick. We're screen sharing. I think it's supposed to open tonight or this afternoon at noon.
We do.
Be open for the next 2 weeks.
We don't need to take a bath with that. We do.
Somebody who's not muted, and I'm gonna, I'll mute them here. Okay. Let's see here. This is our problem. Okay, sign in. So yes, today's is going to open. This one that covers today's lab and last week's lab is going to open this afternoon at noon, and it'll be open for the next 2 weeks. I think it's a good idea for lab to do it that way.
Will that first lab quest still be open today yet or no?
I can extend it out. Give me 1 second. I'll extend it out through today.
Okay, because I didn't realize how bad I did till I just checked the grades. I thought it was something that was going to take time to grade, but
It automatically grades.
Okay.
But give me 1 second here. Edit assignment. Okay. And this one, I think, if I remember right, because I set it up a week ago. Bear with me just for a second here. Continue. I'll extend it out through Friday. Or anybody who's not taken it.
Thank you.
Welcome. I'm going to look really quickly while we're here, and you can see it too. So the second lab quest. It's the same thing. So it's going to be 17 questions. You'll have 40 minutes. Eight matching, matching or labeling, and 9 multiple choice single point. All right, let's get started. Okay. So 1 virtual lab this week. With this lab. So I know that where are you at in lecture? Are you on the lymph system?
Yes.
Okay. So we will do lymph next week in lab. Let's finish up the heart here. What was the pathway of blood through the pathway of blood flow through the heart? I know you guys are all pros at this point. How does it work?
It enters through the vena cava, and then it goes into the right atrium, into the right ventricle, out the shoot, I'm blanking, but out of that one.
Pulmonary trunk.
Okay.
Yeah.
Okay. Where does it go from there?
The lungs.
Okay.
And then it comes back out through the those
What are they called?
pulmonary arteries.
Vein.
It's the reverse, pulmonary veins.
Pulmonary veins.
Okay. And where does it go?
Left atrium, left ventricle, aorta.
Okay. So now today, talking about the electrical conduction of the heart and contraction. Do you remember these terms? What do they mean?
Deep polarization, it means has potential to activate potential.
But yep, the charge starts to flow. Which one charges? Which one starts to flow here?
Depolarization.
Well, which ion forms?
Sodium and
Sodium, correct.
yes.
And we have, in the case of cardiac muscle, we also have calcium flowing, which extends it out. Difference between cardiac and skeletal muscle. What repolarizes? Potassium,
Potassium, the end of the contraction.
right. What defines systole?
Sodium go out, I think, and come potassium inside.
Right. What define the word systole? What is that?
The muscle contracts and then the blood pumps.
Then we have diastole. What is diastole?
The heart.
Relax.
Okay. So moving on into the conduction system, starts in the sinoatrial node. Is the sinoatrial node the only place that has autorhythmic cells that can stimulate a contraction?
Doesn't the AV one also have
The
it?
AV does also. And I'm trying to remember, I think it's the bundle of His branches also can. But the sinoatrial node is the fastest. And that's the reason that it's the one that maintains it. So autorhythmic cells, it's really, really cool. I learned this when I worked with chick embryos, and they were really, really young. So they were, if I had realized what I was looking at, and I kind of knew what I was looking at, but it was 3 in the morning, and I was getting ready to take a plane from Idaho to Arizona for a funeral. And I would have, if I hadn't have been that brain dead, I would have recorded what I was seeing because it was that neat. but I was working with these little chick embryos, and they were really, really, really young chick embryos. Like this is probably about 2 weeks gestation for humans. And I had cleaned it out. When chick embryos develop, they develop on top of the yolk sac. And so what I did was I would manipulate them before this, let them grow for a little bit longer. And then I would cut them out, clean them up, and fix them to section them later. And I had this little embryo that I cleaned up, and I'd gotten all the yolk off it, and I was getting ready to cut it out, and I looked down, and it's beating against the plexiglass. And mind you, remember this is 3 in the morning, and I'm in the lab at school trying to get ready and I've got my bags packed out in my car because I have to go from Arizona to Idaho, from Idaho to Arizona for a funeral. But I'm looking at this little thing as it's beating against the plastic of the plexi dish, petri dish that I'm cleaning it in. And the neatest thing about that, now that I wish I'd really recorded it, is that it's saying that the heart doesn't require any neural, it doesn't require any neural feedback or signaling for it to beat. And it just sat there, it's this new embryo that's about 2 weeks gestation. The heart has just started beating, and it's beating against the plastic petri dish. And I mind you, if I had realized what I was looking at rather than just thinking, oh, that's cool, I would have recorded it. But for me, it was probably one of the neatest things I probably saw in my graduate degree, was that it would sit there and beat against the plastic, even though I've cut it out, and there's no neural feedback going into it. So it starts in the sinoatrial node. Depolarization spreads at that point through both atria. And then it reaches the atrioventricular node, where it pauses for a moment. The fibroskeleton, it takes it a minute to transfer from the sinoatrial node into the bundle of His. This is really important when it comes to filling of the chambers, for it to pause for a second. But it pauses, spreads down the bundle of His, down the right and left bundles, and enters the Purkinje fibers on the right and left side. I think that is the full answer for activity 1, or figure 28.1, is to map that out. Am I right? Okay.
Check it.
I just looked at it too, so I'm pretty sure that's right. So as it spreads, as the electrical conduction spreads, this is the depolarization of the atria. This is the P wave on the ECG. Then we have this gap right here, and then the QRS wave is the depolarization of the ventricles. This gap right here, or right here, represents this move from here down through here. Actually, let's say, I'm going to go back, I'm going to re-say that differently. This gap right here represents this move right here, moving from the sinoatrial node into the bundle of His. This PQRS wave is this depolarization. This is important to be able to assess how this works and where it occurs, where the electrical impulses are for each part of this electrocardiogram is important, because if there is an abnormality in this, you can detect where the problem is in the charge, pathway of charge. So for example, if we find that this distance right here is abnormally large, we know that it's a problem right in here with regards to the pathway of the charge moving. Is that clear? So then repolarization of the atria occurs during this time. It gets lost in the magnitude of this peak. And repolarization of the ventricles occurs right here. So P wave is atrial depolarization. QRS complex or wave is ventricular depolarization. I'm gonna move this. Maybe the PR interval is the conduction, signal conduction through the AV node before activating both ventricles. So this movement right here. The QT is the duration that it takes for depolarization of the ventricles. Or for it to start. The QRS interval is atrial depolarization and diastole, so relaxation of the ventricles, and repolarization, which is concealed by the QRS complex. The PQ sorry, as my brain dies, is signal conduction from the SA node to the AV node. This is what we kind of said before. The ST, ventricular systole and ejection of the blood. So contraction of the ventricles and ejection of the blood. I'm looking at the paper here. That is essentially activity figure 28.3. Any questions? We're going through it fairly fast this morning, if you can't tell. So P-wave depolarization of the ventricles. It's complete after the P-wave. Ventricular depolarization is the beginning of the QRS wave complex. Ventricular depolarization is complete. Contraction occurs and relaxation. And the heart is ready for the next cycle. The reason this is so crucial is when there's issues when there's abnormalities. It's really the only way that we can measure how well the heart is contracting and relaxing, and if it's doing it in rhythm, is by tracking the electrical field conduction and extrapolating what the heart is doing. I think the most interesting, when I was first teaching anatomy and physiology labs, I had a student who had, I would say, his heart was very similar. He had a very enormous, I'm trying to remember what he had. I think he had a really enormous P wave, a small QRS complex, and a small T wave. But he had this enormous P wave. And I'm looking at this, and the students are doing this in the lab, which is what we would do if we could. And they're doing it in the lab, and I'm looking at this, and I'm trying to figure out what in the world is going on. And I'm making sure the electrodes are right. I'm making sure that everything is hooked up right, and it just keeps going. And he's got this enormous P wave, and the rest of the picture is just kind of very small in comparison. And he's sitting there, and as I sit there and I look at it and I look at it, and I'm kind of looking this way and looking that way, trying to figure this out. He finally smiles and he says, "I have congestive heart failure." And I was like, oh, and for me, because I'm a scientist, I'm not a physician, so I don't have to worry about the overall health of the patient. My first statement back to him was, I said, "That's really cool." And because that's what you can do when you're not so worried about the health of patients. But he had congestive heart failure, and that's why he had this enormous P wave. But then his ventricles were slowly giving out.
Talking about P waves, I don't know if anybody caught it, but one of the questions in the lab or the lecture exam, she was talking about P waves and being in AFib, but there is no P waves when you're in AFib. I don't know what the answer is because she doesn't share the answers, but everyone that I've talked to at work says that was the right answer, but her question just wasn't worded very well.
Do you have to, Mom? If you
I
are
can
in,
give you the question if you want to know what it was.
if you're in atrial fibrillation, do you have a P wave at all in the EKG? Because it means you don't have the rhythm. And
No,
okay, see, you're right.
no, because of the atria is quivering.
Yeah, it's not doing any.
It's not deep.
It's not doing any concerted. It's not doing any. What's the word for it? Synchronized contraction.
Yeah, no coordinated contraction.
Okay. Any questions about this? Which one is more dangerous, atrial fibrillation or ventricular fibrillation? If you don't know, take a guess.
VFib.
Why?
Your heart is contracting at a much faster rate than it would if it was an AFib.
True, or it doesn't have any. AFib meaning it doesn't have any. It doesn't have any synchronized contraction of the ventricles.
Yeah.
Let's look at it the way when you talk about fibrillation. You know, we like the fact that heart muscle has intercalated discs. That's kind of important with the heart because we need it to all contract at one time. We need it to all relax at one time. Well, what if they're all contracting and relaxing, but they're not doing it in a synchronized fashion? That's fibrillation. And if they're not doing that, they're not pumping the blood. Go ahead.
Next time I have them at work, I can get you copies of them if you want like real EKGs. I can just white out the names.
If you get really good ones, let me see them. I would love to see them.
I'll keep my eye out.
But so yeah, well, you're not getting good synchronized contraction and relaxation. That's when you're in fibrillation. The heart is just a quivering mess. It's not doing anything that it needs to be doing. It's not functioning because it's not doing it in any synchronized fashion. That's when they go in and they Mother, don't say they die. What's the word for it? The chargers. What's the chargers that they zap you with?
Defibrillator.
Defibrillator. Seeing if they can't get your heart to stop just for a second so that what does a defibrillator do?
Stops the heart to try to get back in the right rhythm.
It stops it just long enough to see if the SA node won't take over again. You guys, do we have any questions about EKGs? I'm trying to think if we have anything. How would you use an ECG to measure heartbeat?
From the beginning of the P wave to the end of the T wave.
You could do it that way, but what's the sharpest indicator on the ECG?
The top of the R wave to the next R wave.
That's how you measure it. If I measure from the very tip of this to the very tip of this, I will get it every time. Because I can't, honestly, I have to be really careful. At what point do we consider this a flat line? At what point do we consider this a flat line? And we have to be very, if we've got to be very particular as to where we're going to measure it from. That's why you do it from the tip of the arteries. What creates the heart sounds?
Is it the opening and closing of the valves?
The opening
Blood
and
rushing
closing
through?
of the valves. And is it the opening and the closing of the valves, or is it the blood kind of pull is it the turbulent blood flow against the valves? For the longest time, I thought that it was the opening and closing of the valves. It is not. It's not like a smack and a smack. It's like a lub, dub. That's a turbulent blood flow. It's not the slapping of valves. They slap close. They open and then they slap close again. That's if you listen to a heart, does that sound like what it is, or does it sound like possibly a pooling or like water rushing into a valve? I think your book says that it's the blood turbulating through that valve, flushing up and moving up against that valve, kind of like the running of water into a cup. It tries to backflow into that valve, and that's the sound that you hear. That's why when you hear a murmur, it's a blood leaking through one of those valves. Mom, does that sound right? That it's kind of a leaking of through one of the valves. Is that right? She says yes, that's right. But when you're hearing the sound itself, yes, is it what you're hearing is the blood flowing into the valve, right? The turbulence as the blood flows into the valve. She doesn't think so. I'm going to look at your book really quick, because I thought I read this just last night, so I'm going to go back and relook, because I don't want to tell you wrong. Heart sounds. Nope, I don't think it's the popping open, Mom. I'm almost sure it's not the popping open. When we listening to the valves made by the heart is called auscultation. Your book doesn't say specifically what they are. No, it does. But no. Oh, so if you look very end, page 710 of your book, very end about heart sounds. The S1 and S2 occur in conjunction with the closing of the valves as a result of the turbulence of the blood in the bloodstream and movements of the heart wall.
Yeah. Snapping shut of the valve.
And then the blood rushing up against it.
No, well, to close it.
You're sure it's not?
Open for the blood atria into the ventricle. When it snaps shut, it's the blood,
Yeah,
the heart contracting to push it out the next.
but what you're hearing is the turbulence of the blood flow. Why
You're
does
hearing
it
the
say that
valve.
right there, Mom?
Does it read it?
But S1 and S2 occur in conjunction with the closing of the valves as a result of the turbulence in the bloodstream and movements of the heart wall.
But that's why the valve closes.
Right.
Not because the blood is turbulent. It has to do with the pressure of the blood against the valve snapping it shut.
Pressure of the valve snapping it shut. Okay. You guys get that answer? So S1 and S2 are the pressure of the valve closing. Pressure of the blood closing the valve. For how many times I've taught that, I realized I was teaching it wrong, and now I'm learning that I was probably teaching it a little right. Let's see here. Let's go forward. This great diagram. How many of you looked at this diagram in your book? Did she show it in class?
I've never seen that before.
It is one of the very last pages right before electrical conduction of the heart. And every time I look at this, I almost have a heart attack because it's a little scary. It is called, bear with me here. I'm going to see if I can't enlarge this. It is called the Wiggers diagram. And it is, and I'll bet you she doesn't talk about it in lecture because it is kind of scary looking. Look at it one at a time. Look at one graph at a time. And it isn't so scary looking. It is on page 711 of your textbook, and it's called the Wiggers diagram. What it relates together is it's relating ventricular pressure, the ventricular blood pressure, the aortic blood pressure, with the filling of the ventricles, with the ECG graph, with the heart sounds. You're going to see this in every physiology textbook you have, because this is like the world famous diagram of heart function. I'm going to try to explain it to you, because I promise you're going to see it over and over and over and over again till the end of time. If you look at it, here we have the first sound right here. We're gonna go down here to where the heart sounds are. We have rapid filling. What is occurring? What happens with the first heart sound? Somebody tell me, what happens with the first heart sound? I'm going to take some Excedrin just a sec. I am not having one of my better mornings, so give me one moment to take some Excedrin to see if I can't get my head to stop hurting. what is happening in that first heart sound? You know, somebody told me what happens with that first heart sound.
Are you talking about S3 that's down there at the bottom?
Is that what that says? Does this say S3 right there?
In the blue, it says S3.
Is this S2? Is that what that says?
Yeah, that's S2.
Okay, let's go with S2. What's S2?
Okay. S2 is isovolumetric relaxation. So that's when all of the valves are closed.
That's all the valves are closed. Okay. So what is happening in the heart at that moment? Actually, no, it's right here. Look, right about right here to right here. What is happening right there? Right before S3. What is happening? Are all the valves open or are they all closed?
The ventricles are filling, so it's just the tricuspid and bicuspid that are open.
They're open.
Right.
So they're open. So right here, this is the ventricular pressure right here. As the ventricles fill, what is going to happen to the pressure in the ventricles? Not a whole lot. It's going to kind of increase, but it's not really going to increase until we contract the muscle, right? So it's kind of relaxed and it's moving along. And this graph right here is the ventricles are filling. This is the blood volume in the ventricles. They're filling a little bit. Right about right here, what happens right here in the P wave, guys?
SA node contraction.
It's the SA node contraction. Well, what does that result? How does that relate to overall heart function?
Start atrial contraction.
It's atrial contraction. So all of a sudden, right here on this graph up here, which this black line is the measure of ventricular pressure, all of a sudden the pressure is going up. Why is the pressure going up right there in the ventricles?
The AV valve closes.
The AV valve is going to close here in a second, but this is right now the atrial contracting, and that's pushing the last little bit of blood into the ventricles. Then this is why we see this little tiny spurt in the blood pressure of the blood pressure of the ventricles. We also see right here, we see the blood volume push up just a little further in the ventricles. Most of blood filling of the ventricles occurs just because of fluid flow as a result of gravity. The last 30% of filling right here is because of atrial contraction. Which occurs right here and right here. So we're right here. This is atrial contraction, atrial systole, right here. At atrial systole, what happens at the very end of atrial systole? What's this heart sound right here? The valves closing of the bicuspid and the tricuspid and the bicuspid valves are closing, right? The atrioventricular valves are closing. Atrioventricular valves are closing. What is happening to the blood pressure in the ventricles at that point? If the atrioventricular valves are closing, what's the blood pressure look like in the ventricles?
Increasing.
Increase.
It's massive. It's going through this jump right here. It's massive. Where is that blood going? It's going into the aorta and the pulmonary trunk. So at the same time that the blood pressure is elevating in the ventricles, it's also elevating in the aorta. The black line is the ventricles. The red is the aorta.
So I understand a lot of this diagram. I'm just struggling with the red lines. So the aorta, as in, the part above the heart, the artery, but what is the left atrial pressure? Is that
the left atrial pressure is the pressure in the left atrium.
the left atrium? So why is that one? I guess what I'm struggling with is why are those two?
It doesn't ever have a lot of pressure in it. Why do they put it in there? I don't know.
Okay, yeah, that's what I'm confused.
I think the only reason they put it in there is so that you know what's going on with the AV valves.
Okay.
This is a good question. That's the reason they put it in there.
Mm-hmm. Okay. Yeah, because I was just struggling to understand why it was necessary, but that makes sense then to do the AV valves.
Well, you know what, Jordan, you're years ahead of me if you understand that much already. Because I've been looking at this diagram for several years, and every time I look at it, I kind of have a heart attack. But I'm realizing that if you start breaking it down, it makes sense. But you have to not look at it like I do sometimes and realize, okay, that's a lot of data. Start breaking it down and looking at the individual pathways and you see it. So right here, the volume is going up. It's peaking right up here. About right here, the volume in the ventricles, the pressure is peaking, the volume is peaking. This is your end systolic volume right there. What happens at that point? When the volume is peaking in the ventricles, or excuse me, the pressure is peaking in the ventricles, the volume is peaking there. You're at the max. That is when the pressure in the ventricles is going to exceed that in the aorta and the pulmonary trunk. And you're going to be able to open the semilunar valves. You can't open those valves until the pressure inside the ventricles exceeds that in the aorta and the pulmonary trunk. So you open it, all the blood rushes out of the ventricles. The pressure in the ventricles drops. Drops. And right here, you're at the end diastolic volume in the ventricles. This is after all the blood is has left the ventricles. And we hear that the second heart second heart sound. And we have what's called the dichrotic notch because the valve's close. The semilunar valve's close. And then it starts all over again. If you can break this down and understand how it relates to heart function, there's not a whole lot about the heart that you don't understand at this point. Isovolumetric contraction, what does that mean? It's isovolumetric contraction. The ventricles are contracting, but all four valves are closed. So the volume is staying the blood volume is staying the same. I miss this. You gonna get over here? Come on. Come on. Blood is released right here. Blood is leaving the ventricles. Our blood is relieving the ventricles. And we're now going to reach our end systolic volume. Then isovolumetric relaxation. Blood starts filling the ventricles again. Any questions? Okay. So what is the end diastolic volume? How do you define that term?
Would that be the blood volume in the ventricle after it's contracted?
After it's contracted, how about we say before it contracts? Let's say it's the blood volume before it contracts. So this is at the very end. Diastolic is the relaxation. So this is the volume in the blood after its maximal amount that it's filled with. So it's been engorged with blood. That's the end diastolic volume. What's the end systolic volume? What's left in the ventricles after it contracts. So endostolic, what's left there when it's at the end of its relaxation. And it's just filled up with blood, and it's kind of just filled up with blood. I'm gonna stop sharing my screen for a minute. I'm just gonna try to explain this using my hands. Bear with me just for a second here. So hopefully you just see my face here. So end diastolic, we have our heart here, ventricles right, atria right here, ventricles right here. The blood starts filling the ventricles, they start kind of moving out. Mom, would you grab me one of my balloons really quick before you leave? Yeah. And my students will watch how well I blow up a balloon. No, I can blow it up. I've never had a lot of lung power, so whenever I have balloons that I need to have filled up, I let someone else do it. It doesn't matter. Okay. Okay. Have fun buying a new clothes washer. It will be delightful. Okay. So this is end of diastole. End of diastole. All the blood is left. Oh, sorry, end of systole. Wrong one. End of systole. All the blood is left. I'm going to start in atrial systole. Going to put a little bit of air in there. Okay, so a little bit of blood in there as I get lightheaded because I can't blow up balloons worth anything, but filled it up a little bit. Now we're going to have atrial systole. Atrial